Hello students, this is molecular basis of inheritance part 11. In this slide we will try to understand how the DNA get packaged inside the cell, which is a beautiful animation. You can understand the different steps which are involved in the packaging of a eukaryotic DNA in this slide. So let's try to understand the structure. Now this thread like structure, you can observe this is nothing but the double helical structure of the DNA. So the magnification you can see. So see how beautifully the double helical structure it gets packaged. Now these are the histone proteins which are getting attached to the DNA. See how beautifully the double helical DNA structure it forms the nucleosome by wrapping around. Now this each nucleosome again you can observe it again many nucleosomes they get coiled inside the cell. This is the chromatin fiber 30 nanometer. Now see how beautifully now this chromatin fiber it again gets supercoiled to form a structure which looks like the chromosome. See how it is getting coiling, supercoiling. Can you observe that? Now it is the chromosome and this is the cell. Here you can observe these are the chromosomes, right? And this is the metaphase. You have studied the cell division, anaphase. You have studied it, right, in your 11th standard. See how beautifully the telophase, which is the last phase, cytokinesis, and from a single cell, the two cells they get formed. So these were the various steps which were involved in the packaging of eukaryotic DNA. Let's try to understand this each and every step one by one. In today's video we are going to discuss about differently how the packaging of genetic material it occurs in case of eukaryotic cell. So you can observe this is the eukaryotic cell structure. This is nucleus. This part is the nuclear envelope. These are the nuclear pores. These are the ribosomes which are situated. Right. This is smooth endoplasmic reticulum. This is a rough endoplasmic reticulum. You have studied this all these structures in detail in your 11th standard. Now these are the lysosomes, plasma membrane, right? So these all things you have studied. Now this is the general idea about how an eukaryotic cell it looks like. Okay, so in the eukaryotes they have well organized nucleus. You have studied in the previous lecture that in case of prokaryotes, the prokaryotic cell they don't have such compartments, right? uh, so in case of eukaryotes, there are some differences between the eukaryotic cell and the prokaryotic cell. So eukaryotic cell, it contains membrane-bound organelles, like you can observe in this diagram that there are number of organelles which are present inside the cell and they are membrane-bound, including the nucleus. Nucleus is having a covering that is called as nuclear envelope. So it is again membrane-bound structure. So eukaryotes can be single-celled or multi-celled, as such as you, me, plants, fungi, and many insects. These are the examples of eukaryotic cell. And prokaryotic cells, they do not have or they do not contain the nucleus or any other membrane-bound organelle. So such membranes or such compartments are completely absent in case of prokaryotic cells, while in case of eukaryotic cells all such compartments they are present, as the structure of eukaryotic cell is more complex, more complicated than the prokaryotic cell. So they contain nuclear membrane, nucleolus, and the thread-like material in the form of chromosomes. So their genetic material is present in the form of thread-like structure. How it is packaged that we are going to discuss in the next slides. So in this slide you can observe this diagram where the chromosomal structure is shown. The chromosome is present. The genetic material in the eukaryotic cell, it is packaged in the form of chromosomes. Now you can observe this chromosome, which is highly condensed, duplicated chromosome, which is present in the dividing cell. Okay, these are the chromatids, and this is called as heterochromatin. We are going to discuss this part, what is heterochromatin, what is the Euchromatin, later on. Just try to understand this diagram. So let's try to understand that how a double helical structure, that is, now this diagram is nothing but this is the double DNA helical structure, which is seen through the microscope, which we can observe that this, it looks like a thread-like structure, right? So this thread-like genetic material is packaged. Now how it is packaged in the eukaryotic cell, that we are going to discuss in this slide. So this is the double helical structure of DNA, and in the next part you can observe, now this is the part which is zoomed in. Okay, so this segment, this square, if you are able to observe on your screen, so this is the square part which is shown in this diagram, that is the DNA helical structure. So the nucleosomes which occur or which appear like beads on string. Now why it is called as beads on string, you can observe this diagram. It looks like beads on string. These are the dots which appear like beads, and the thread-like structure is called as the string. Okay, so they form these bead-like structures. These are nothing but the nucleosome beads. Okay, these are termed as nucleosome beads. These are the histone proteins. This each circular bubble-like structure or circular structure, it is nothing but the histone protein. Okay, so the DNA is wrapped around on these nucleosome structure. Now this thread, you can observe, this is different in color. Its color is light, right? So this thread-like structure is wrapped around these nucleosome structures. Okay, so this is about the beads on string structure. Now just again observe that how it gets again compactly packed. Okay, now a thread, we have started with a long thread. Now the thread is wrapped around firstly on the nucleosomes. Now these nucleosomes, they are again compactly they get arranged, so that is shown in this diagram. Now these are the different kinds of nucleosome proteins which again get compactly arranged in the next structure. So these are the nucleosomes. Now see, you can observe the difference between this thread-like structure and this is the nucleosomes which are compactly arranged in the diagram, right? So these are the DNA nucleosomes which are compactly arranged. Now this is the loop which is shown. This part is nothing but this segment which is zoomed in. Okay, so it is present in the form of looped domains. You can observe, now this is the looped domain. Okay, so these histone proteins, they are actually present in the form of such loops. Now this is again shown that how it is coiled and supercoiled. Now this is supercoiling, and again it is supercoiled or arranged in the form of in the chromosomes. If you're able to understand that from a thread-like structure, we are arranging the or we are compactly coiling or supercoiling the DNA or the genetic material to so that it should get fit in the eukaryotic cell. Okay, so DNA is associated with histone and the non-histone protein. So it is DNA is actually associated with two kinds of proteins. First is the histone protein, and another are the non-histone proteins. So this proteins, they were reported by R. Kornberg in 1974. The organization of DNA is much more complex in the eukaryote. So you can observe the complexity. We can understand, right? It is little complicated to understand even. So histones, they are the proteins that are rich in lysine and arginine residues. Both these, now these names are very important. If the question is asked on the packaging of eukaryotic DNA, then of course you have to mention the histone and non-histone protein. You have to mention the complexity of the DNA organization in the eukaryotic cell, and of course you have to write something about the, or you have to mention the names of the histone proteins, that are lysine and arginine residues. Now, why the mentioning is important? Because these are the amino acid residues which carry the positive charge on them, and DNA is negatively charged. So you can just imagine when the two different kinds of charges, when they come together, DNA is negatively charged and the histone proteins, they are positively charged. So when the coiling is occurring, so for example, my fist is the histone protein, and if the thread is the negatively charged DNA, and if I'm coiling it, then that attachment will be a much powerful, right? Because the negatively charged DNA will get more attracted towards the positively charged nucleosome. Okay, and because of that, the this type of supercoiling or coiling is a very very attractive or very powerful. So in this diagram, or in this slide, we are going to discuss about the histone proteins. What are the histone proteins? Now this is the histone protein, octamer of core histones. Octa means 8. Okay. So 1, 2, 3, 4, such, again 1, 2, 3 and 4, such 4 proteins, they core proteins, they are present, and they are compactly arranged, and they form their set, it forms the histone protein. So they are named as H2A, H2B, H3 and H4. This is a red colored structure. You can observe this is called, this is the core DNA, which is coiled or which is wrapped around the histone proteins. Okay, now this is H1 histone and this is the linker DNA. So let's try to understand what is the histone protein now. Histones are a set of positively charged basic proteins that are histones and protamine. So they are positively charged, as I have already mentioned earlier, that histone proteins, they are positively charged, and the DNA which is getting wrapped around the histone proteins, it is negatively charged. So these histones, they organize themselves to make a unit of 8 molecules. As the 8 molecules, the unit of 8 molecules, they are present in the histone proteins, and because of their organization, it is called as histone octamer. Octa means 8. So the negatively charged helical DNA is wrapped around the positively charged histone octamer. This structure is called as nucleosome. Now you can get a question like explain the nucleosome, or you can get a question like write a short note on nucleosome. Then of course you have to draw the diagram, and you have to write the positively charged basic proteins, or you have to write about the histone octamer. You have to specify the histone octamer and its charge, and the structure, whole structure is called as nucleosome, and the nucleosome core is made up of 2 molecules of each 4 types of histone protein. These are H2A, H2B, H3 and H4. Of course the diagram is important if you are going to write all the points. Okay, so that's all about the histone structure. In this slide, you will be able to understand that how many base pairs they get wrapped around. So how many part of the DNA it gets wrapped around on one nucleosome, and how the such a long DNA or deoxyribonucleic acid, it gets wrapped and coiled and supercoiled to form a structure like chromosomes. So these diagrams are more colorful, so you can observe the diagram. One nucleosome approximately contains 200 base pair long DNA helix, which is wound around it. So about 146 base pair long segment of DNA remains present in each nucleosome. So you can understand the base pair structures of the chromosome. Now this is the double helical structure. It, the one helix, one part, now this one segment, you can say, it contains 10 base pairs. It measures 3.4 nanometer. This is 2.4 nanometer. This is the duplex DNA, the total length of the DNA here you are talking about. Now see, the nucleosome core, it 146 base pair, it gets wrapped around the DNA histone core. Now this wrapped around part, the DNA segment which is wrapped around or which is coiled on the nucleosome, that segment you are talking about, it is near about, it contains 146 base pairs. Again, now this next part is the chromatin, which is the extended base on string structure. We have already discussed this part. So this one part you're talking about, your this length is about, or this size is 12 nanometer. This is the linker DNA, 60 base pairs. This wrapped DNA, it is, of course, we have just mentioned that it contains 146 base pairs. And this is compact chromatin or hypothetical structure of the DNA. And in metaphase, the chromosome, you can see this 1500 nanometer, and this part or this segment is 30 nanometer. Okay, so here about 146 base pair long segment of DNA remains present in each nucleosome. What is a nucleosome? These are the repeating units of chromatin, which are thread-like stained or colored bodies present in the nucleus. So here we are discussing the how many base pairs of the DNA they get coiled on one nucleosome structure. So in this slide you can observe that beads on string structure clearly. This looks like beads on string when observed under an electron microscope. Now you can observe in this, it just looked like this beads on string structure. So six such nucleosomes get coiled and then form a solenoid that looks like coiled telephone wire. So this is the nucleosome position sliding 10 nanometer, and under electron microscope it exactly appears like the beads on string structure. In this slide, we will try to understand whatever we have studied, that how the nucleosome get formed by wrapping, coiling, supercoiling of the DNA, how the chromosomes they are formed by the coiling of the DNA structure, how the negatively charged DNA is wrapped around the nucleosome. That all part we will try to understand with the help of animation in this slide. So this is the heritable modification. You can observe we are going inside the cell. Now this structure is the cell. In this cell, eukaryotic cell, this is the nucleus. The nucleus, we know that nucleus contains the genetic material. And in case of eukaryotic cell, if you are talking about, the genetic material is compactly arranged in the form of such chromosomes. So you can observe these are the chromosomes. Now we are going inside the chromosome. Now this structure is called as chromatin, a thread-like structure. Now we are again going in that chromatin structure. You can observe these are the nucleosomes. See how many nucleosomes they are compactly arranged in the chromatin fiber or thread. Now this is the nucleosome. You can observe now this structure is the negatively charged DNA. Okay, and these are H2A, H2B, H3 and H4. These are the histone proteins. Can you see that? These are the octamer structures. So this nucleosome is present and this negatively charged DNA is coiled around the histone protein. Okay, so there are two kinds of proteins which act or which play very important role. They are HAT and HDAC. So histone acetylation and deacetylation is the phenomenon which, or the processes which are carried out by these two proteins. And these are very important processes by which the lysine residues with the N-terminal tail protruding from the histone core of the nucleosome are acetylated, and the deacetylation, so there is acetylation and deacetylation, it occurs by these two proteins. These are HAT, that is called as acetyl transferase, and another is HDAC, that is histone deacetylase. So histone acetylation and deacetylation is very important, or they are very essential part of the gene regulation. these reactions, that is acetylation and deacetylation, these two reactions, they are typically catalyzed by the enzymes which I have just mentioned, that HAT and HDAC. So acetylation is a process where an acetyl functioning group is transferred from one molecule to another, while deacetylation, it's simply the reverse reaction where an acetyl group is removed from the molecule. So that you have just see observed in the animation, right? So acetylation and deacetylation, here you're talking about as gene expression. As I have told you that the nucleosome structure is positively charged, on which the DNA molecule is wrapped around. So when there is gene expression, when you talk about the expression of gene or formation of protein, so for that process the deacetylation is very essential. So the HAT and HDAC, they are very important as far as the gene expression is concerned about such condensed or supercoiled or compactly arranged DNA, if you are talking about. Okay, so the chromatin is packed to form a solenoid structure of 30 nanometer diameter, and further supercoiling tends to form a loop structure that is called as chromatin fiber, which you have just observed in the animation, which further coils and condense at the metaphase stage to form the chromosome. So you can just observe this animation, which is a beautiful and amazing, I mean, animation where you can just go inside the cell, and in that cell you can observe that in the nucleus how the in the chromosome, how the DNA, it is compactly arranged. Okay, so that's all about this. And the names of the HAT and HDAC, uh, this is actually, it is not given in your 12th book, but of course the as far as your need is concerned, such questions can be asked, and this part is again very important. So in this slide we will try to understand what is the difference between heterochromatin and euchromatin. So this is the chromosome. You can observe the color of the chromosome, the bands you can see, the dark color and the light color, the color differentiation you can observe. The dark color which you can see in the chromosome, that part is nothing but heterochromatin, while the lighter part, that band or that portion, is called as euchromatin. So heterochromatin, it is more condensed, and because of that, when it is stained, so it observe light dark in color. The genes, they are methylated, they are silenced, means these genes, they do not get expressed. Poor genes are present, the content is high, adenine and thymine content is present in high amount or a large amount. The stain, it stains darker when it is observed. Euchromatin, now this is the lighter color band. So these genes, they are less condensed. These genes which are present in this segment, they are expressing genes, so they are not silenced, they are expressed, they show their expression. They are, this part is rich in the GC content, that is guanosine and cytosine. It is high in amount and it stains lighter. Let's try to understand what is heterochromatin and euchromatin in the form of differentiation or in the form of the table. In this slide, we will try to understand the structural differences, the difference between the heterochromatin and euchromatin. Heterochromatin, in eukaryotic cells, some segments of the chromosome during interphase and early prophase remain in a condensed state, constitute heterochromatin. And euchromatin, these are the regions of chromosomes which are in non-condensed state and constitute euchromatin. So it is genetically mostly inactive, heterochromatin, while euchromatin is genetically very much active and it is fast replicating. Heterochromatin, it stains strongly and appears dark, while euchromatin, it stains light. Heterochromatin, it is 2 to 3 times more rich in DNA, while euchromatin, it contains less amount of DNA. Heterochromatin, it is transcriptionally inactive, while euchromatin, it is transcriptionally active. That's all about today's session. Thank you.